Dear students, Assalamu alaikum. The objectives of today's lecture are to learn about pre-processing of lymphocytes and secondly, the lymphocyte clones. Pre-processing of the lymphocytes. In the last lecture, I mentioned that for the lymphocytes to become fully functional, they have to be pre-processed before that, and all of the lymphocytes eventually end up in the lymphoid tissue, but before doing so, they are pre-processed. Now you can see in this figure, which is taken from the textbook of Guyton, that all the lymphocytes, they take their origin from the common lymphoid progenitor cell, and they develop into the T lymphocytes or the B lymphocytes. So the T lymphocytes, they are first processed in the thymus, where this pre-processing takes place, and the B lymphocytes, they are pre-processed elsewhere, as I will mention later in the lecture. So let's proceed with this process of pre-processing. Now we can divide this pre-processing of lymphocytes into two stages. Number one, non-antigenic development, and secondly, the antigenic development. By non-antigenic development, I mean that the lymphocytes, they don't come in contact with any antigen till this non-antigenic development is completed. They are processed by the body tissues, and there are certain capabilities and capacities which are developed in these lymphocytes. So what they do is that firstly, the lymphocytes, they express antigen receptors before ever encountering the actual antigen. So they first develop the antigen receptors, which are proteins in the membranes, and this occurs before they encounter the actual antigen. Now what happens is that each lymphocyte with its own antigen receptor, which is a special protein, it responds to only a very, very small part of the total universe of antigenic epitopes. So there are millions or billions of antigenic epitopes, molecules, and each lymphocyte, it has the capacity to react to that epitope because it has a corresponding antigenic receptor which has been developed to respond to that antigen epitope. So this means that they are sufficiently diverse to deal with any antigen, meaning that all of the lymphocytes, they are different, each having the capacity to develop, to react to any antigen. For example, lymphocyte A will have the receptor protein A, which will respond to the antigen A. And so let's say a lymphocyte number 1 million will have a different receptor protein to deal with antigen number 1 million. So this is how these lymphocytes, they are different. Now for this to happen, the lymphocytes first migrate to and are pre-processed in the thymus gland. The T lymphocytes I'm talking about, the T lymphocytes, they migrate to the thymus gland for this process to occur. Now, what happens in the thymus? They divide rapidly, and then they develop this diversity against different antigens, and I have just tried to explain to you that what is the meaning of this diversity. And keep in mind that the diversity results from specificity. So what is the specificity? Specificity is that one thymic lymphocyte, it develops specific reactivity against one antigen, and the other lymphocyte develops specificity against another antigen, as I mentioned, the number 1, 2, 3, 4. So each lymphocyte will react with a specific antigen, and this is known as the specificity. Okay, next, now this, as I have already mentioned, this specificity is a property of an antigen-specific receptor protein present in the cell membrane of that lymphocyte. Now, each lymphocyte has the potential to make millions of different antigen-specific receptors, meaning it has the capacity or capability to make any of millions of receptor protein, because it has the DNA, and DNA can make any of amongst the millions of a specific receptor protein. Now, what happens? That lymphocyte, it randomly selects a unique DNA arrangement that encodes a unique receptor protein. So simply, each lymphocyte will make, although it has a choice to make millions of receptor proteins, but it will randomly select a unique DNA arrangement to encode or to make a unique receptor protein, and that is the receptor. Now, all daughter lymphocytes which will arise from that lymphocyte, let's say lymphocyte B, will produce the same antigen receptor protein, and therefore all these daughter lymphocytes which take their origin from a specific lymphocyte, let's say lymphocyte B, they are called the clone of that lymphocyte, and I'll come back to this clone formation a little while later. The process of diversity continues until there are thousands of lymphocytes with specificity against thousands or even millions of different antigens. So this process continues, and this is, mind you, occurring in the thymus for the T lymphocytes. Now, in addition to this, developing the diversity and the specificity, in addition to this, a very important function is that the thymus also makes certain that any of the T lymphocyte will not react against the self proteins or the self antigens. So this again is a very important function, and this is called the immune tolerance. And how it does is that the thymus selects which T lymphocytes will be finally released after pre-processing by first mixing them with all the specific self-antigens of the body's own tissues. So it kind of gives them the chance to mix with the self-antigens of the body's own tissues, and then it identifies those lymphocytes which react. So after identification, if a certain T lymphocyte is reacting, it is destroyed and phagocytized instead of being released in the blood. Now this Mind you, this happens to up to 90% of the cells, which is a very, very large percentage. So all those lymphocytes which react with these self-antigens, they are destroyed. They're not allowed to reproduce, and this is known as immune tolerance. And if this process is somewhere deficient, this leads to autoimmunity, because those T lymphocytes, they are not properly dealt with, they are not destroyed, and they enter the body and they can destroy the self-antigens. And we'll study this in the next lecture. Now, after this pre-processing has taken place, now the pre-processed T lymphocytes, they leave the thymus, and they get lodged in lymphoid tissue everywhere. And we know where is the lymphoid tissue: in the submucosal areas, in the spleen, liver, lymph nodes. So they get themselves lodged there after pre-processing. Now, this pre-processing of the T lymphocytes in the thymus, it is occurring shortly before the birth of the baby, and it continues for a few months after birth. Now, the pre-processing of the B lymphocyte occurs along the same principles and same methods as the T lymphocytes. The only difference is that the B lymphocytes, they are pre-processed in the liver during the mid-fetal life, and in the bone marrow during late fetal life and after birth, instead of the thymus. And because this pre-processing of B lymphocytes was observed first in the bursa fabricius in the birds, so therefore these lymphocytes are termed as, or they were named as, B lymphocytes after the bursa of fabricius, which is an organ in the birds. So the pre-processing of B lymphocytes occur along the same lines and the principles, only the tissues are different, for example in the liver and in the bone marrow, in contrast to the thymus. Now this pre-processing of the B lymphocyte is different from the T lymphocytes in two ways. Firstly, the B lymphocytes, they secrete antibodies, and therefore they have a greater diversity, meaning they have greater receptor protein, number of receptor proteins which can react to a greater number of antigens. And we'll study this in more detail when we'll study the humoral immunity, which is the function of the B lymphocytes. Now similarly, after like the T lymphocytes, after pre-processing, the B lymphocytes, they migrate to lymphoid tissue throughout the body, where they lodge in the germinal centers, and these centers are very near to the groups or aggregations of the T lymphocytes. Now this is the point up to which these lymphocytes, whether B or T, they haven't come across any antigen. They just have developed the capacity and the capability to react to any antigen, but they have as yet not encountered any antigen. They just are waiting there in the lymphoid tissue, in the lymphoid organs, to meet a specific antigen. Now, after they meet this antigen which is specific to them, one of them, then the next stage starts of the pre-processing, and that is known as the antigenic development. And here is when the role of the lymphocyte clones it is started. Now, what is a clone? Basically, what happens is that when specific antigens come in contact with the T and B lymphocytes, as we have just studied, which are lying in the lymphoid tissue. Now, some of the T lymphocytes, they become activated to form activated T cells when they come in contact with a certain antigen. And some of the B lymphocytes become activated to form the antibodies. Now, as we studied earlier, each of the preformed lymphocytes is capable of forming only one type of antibody or one type of T cell. So, once the specific lymphocyte is activated by a specific antigen, it reproduces to form large number of duplicate lymphocytes, which are exactly the same as the parent lymphocyte which has been activated, whether that is a B lymphocyte or whether that is a T lymphocyte. So these duplicate daughter lymphocytes, all of them are similar to each other, and therefore they are known as the clones, the clone of that lymphocyte. So you can see this in this figure, that somewhere here, this is the an X lymphocyte, this is an A lymphocyte, this is a B lymphocyte, this is a Z, and this is the Y lymphocyte. So there is an antigen X which is to which this receptor protein on the X lymphocyte is already there. So it will bind with its receptor protein present on the X lymphocyte, and that X lymphocyte will then become activated, and it will reproduce to form many lymphocytes which are very similar to the X. So all this is known as all these lymphocytes which have developed from the activation of certain lymphocyte. This is known as the clone of that lymphocyte. So when a receptor is activated by the binding of a specific antigen, the lymphocyte repeatedly divides, resulting in a clonal population of daughter lymphocytes which are very similar to the parent lymphocyte. So here again you can see a B lymphocyte, three different B lymphocyte, B1, B2, 3, and then they are activated by a certain antigen, and they form this B2 is activated, it is proliferated and differentiated, and it further forms the antibodies. So all this process is known as the cloning of the lymphocytes. Now the lymphoid tissue, it contains thousands of unique clones of the T and the B lymphocytes, which are able to react to a certain antigen, each of them. So when a specific antigen binds with the precursor or the granddaddy or the grandparent of a specific clone, this results in the stimulation and proliferation of those clones. So this was about the clones. Now the origin of the clones. So how do they take origin? How do they form? How do this baffling number of lymphocytes can be formed? So the thing is that the whole gene for forming each type of T lymph or B cell is never present in the original stem cells, because that would have meant enormous amount of DNA has to be present in the nucleus. So only gene segments are present, and these gene segments during pre-processing of the respective T and B lymphocytes, these gene segments, then they can become mixed with one another to form millions of combinations, and then they can produce millions of receptor proteins on the cell surface. Next is the mechanism of activation of clones. So how they are activated, we have already studied that each clone of lymphocyte is responsive to only a single specific antigen, or maybe to very similar antigens, and therefore an antigen stimulates only those cells that have complementary receptors for that antigen, as we have studied. And in this in the mechanism of activation of clones, the macrophages have a very important role. So what do? The macrophages, they pass the antigens by cell to cell contact directly to the lymphocytes, leading to the activation of specific lymphocyte clones. And we will study this in more detail when we will study the T cell immunity or cell mediated immunity. Another factor is that these macrophages, they secrete interleukin 1 that promotes the growth and reproduction of the specific lymphocytes. So this is the role of the macrophages in the activation of clones. We'll continue with this immunity, and we'll continue with this formation of the beta cells in the next lecture.